Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
We hear so much about common diseases, the ones always in the news. But today, we're focusing our lens somewhere different on a crisis that's often in the shadows. It's a single, devastating neurodegenerative diagnosis. So rare, it hits maybe only 1 in a million people globally. We're doing a deep dive into Infantile Neuroaxonal Dystrophy, IADA, or under its broader classification, PLA2G6 Associated Neurodegeneration, PLAN. This is a really cruel disorder. It essentially robs infants, toddlers, of their ability to move, to speak, often taking their lives within the first 10 years. So our mission for you, listening in, is to unpack the tragedy, yes, but more importantly, the science, the cutting-edge stuff that's trying to fight back. Our material today, it comes mainly from a big review looking at everything molecular, clinical, therapeutic, published between 2020 and 2025. It's dense, but we'll pull out what really matters. This whole thing, this tragedy, it starts with a tiny genetic mistake. So maybe let's begin with the clinical picture. What does that look like for a child, for a family when IND is diagnosed? What's the timeline?
Well, what's so particularly devastating is the contrast. You see, INAD is inherited. It's autosomal recessive, meaning the child gets 2 faulty copies of a specific gene, but it almost always shows up after a period where the baby seems to be developing perfectly normally.
Right.
The onset usually hits somewhere between 6 months and 3 years old. Often it's right around that first birthday mark, 12 months.
Wow, that's such a key time. Walking, first words.
Exactly. They might hit those early milestones and then the decline starts. And it's rapid. It's actually regression.
Regression. So not just stopping development, but going backwards.
Precisely. They actively lose skills they've already mastered. Motor control slips away. Language disappears. Social interaction fades. Initially, parents might note when the baby seems floppy. We call that axial hypertonia. Just poor control over posture, weak trunk muscles.
And how does that change over time? Does the floppiness stay?
No, it shifts pretty dramatically, actually, into progressive spasticity. So severe muscle stiffness, rigidity. And that's often combined with something called cerebellar ataxia, which is a really chaotic lack of coordination, makes purposeful movement almost impossible. Think about a child who should be learning to run, now struggling even just to sit steadily.
That's incredibly difficult to picture.
It is. And alongside all that, you often see major visual problems developing. Optic atrophy, that's the deterioration of the optic nerve, and nystagmus, those involuntary jerky eye movements. Ultimately, this relentless neurological damage affects bulbar function. That's the control center for swallowing and breathing.
Oh, no.
Yeah. And sadly, that often leads to respiratory complications, which are frequently the cause of death, usually within that first decade.
That's horrifying. And you mentioned a pathological hallmark earlier, these neuroaxonal spheroids. You said that like the physical evidence of the breakdown. What exactly are they? Not tumors, right?
No, not tumors at all. Think of them more like, junk piles.
Yeah.
Or maybe roadblocks. They're focal swellings, dilations within the axons, those long nerve fibers that transmit signals. These axons degenerate and these swollen spots appear.
And what's inside these swellings?
When you look closely, they're just packed, clogged really, with disorganized cellular bits and pieces, vesicles, waste material, and significantly a lot of swollen, damaged mitochondria, the cell's energy factories. It tells us the whole internal transport system, the supply chain that keeps the neuron alive and working, has just fundamentally failed. It's completely backed up.
Okay, so let's trace that failure right back to the source. This massive breakdown, this supply chain collapse, it comes down to one gene, right? PLA2G6. On chromosome 22, what does this gene normally do? What's it supposed to build?
It holds the instructions for making a really vital enzyme. It's called phospholipase A2 type cis, or sometimes IPLA2-VIA.
Phospholipase. Okay, break that down. What's its job in plain terms?
Okay, think of it as the cell's primary maintenance and repair specialist, specifically for its membranes. Cell membranes around organelles like mitochondria.
The boundaries, the walls, essentially. And critical for the mitochondria, the power plants.
Exactly right. This enzyme is crucial for keeping those membranes healthy. It helps break down and recycle old or damaged phospholipids. Those are the fatty molecules that make up the membranes. It's constantly turning them over, replacing worn-out parts. So when this enzyme doesn't work properly because of the gene mutations that damage stuff, those worn-out membrane lipids, they just accumulate, they build up.
And that's the junk we see inside the spheroids.
That's a big part of it, yeah. The whole system gets clogged, membrane repair fails, and it contributes directly to that axonal swelling and degeneration.
So if this enzyme is so important for all cell membranes, Why does INAD hit the nervous system so hard and so fast? Why isn't it causing, say, immediate liver failure or kidney failure too?
That's a really great question. It boils down to selective vulnerability. While the enzyme is present in many tissues, its function is absolutely critical in cells with really high energy demands and high membrane turnover. And neurons, especially certain types like those in the cerebellum, which controls coordination, and the basal ganglia, they are incredibly demanding. They need constant maintenance.
Okay.
Plus, this specific phospholipase, PLA2G6, also plays a key role in dealing with oxidative stress. It helps clear out damaged peroxidized fatty acids from membranes.
Oxidative stress, like rust in the system.
Kind of, yeah, a byproduct of high energy use. Neurons face a lot of it. So when the enzyme fails, these high-demand neurons, the ones needing the most cleanup and repair, they're the first and hardest hit. They can't keep up.
So the cell structure breaks down and its energy supply gets compromised all at once.
Wow. And does this also explain why the disease can look different in different people? You mentioned a spectrum. It absolutely does. It correlates really well. The severity of the mutations in the PLA2G6 gene tends to predict the age of onset and how fast it progresses. So if you have severe null mutations, meaning the gene is so damaged it makes basically no functional enzyme at all, that typically leads to classic, severe, early onset INAD.
Right.
But if you have what are called hypomorphic mutations, these still allow for a little bit of residual enzyme activity, even just a tiny amount that often results in later onset and be slower progressing forms. We sometimes call that atypical neuroaxonal dystrophy or AND.
So even a little bit of that maintenance function makes a difference.
A huge difference, potentially delaying onset by years or even decades in some cases.
Okay, moving from the tiny molecular fault to how doctors actually figure this out.
Yeah.
How is INA diagnosed today? What are the key tools?
It really requires putting several pieces together. Neuroimaging, especially an MRI scan of the brain, is absolutely crucial. One of the most consistent early findings doctors look for is cerebellar atrophy. That means the cerebellum, the brain region critical for coordination, is shrinking. And that fits perfectly with the ataxia, the movement problems we see.
Makes sense. What else might they see on the MRI?
Well, there's this connection to a group of disorders called NBIA that stands for neurodegeneration with brain iron accumulation. So, in some, but not all, planned patients, the MRI might show signs of iron building up in specific deep brain areas, like the globus pallidus and substantia nigra. It shows up as low signal intensity on certain MRI sequences.
Iron buildup. That sounds ominous.
It is, and we'll get back to why. But this iron finding can be variable, especially early on in INE. However, there's another more unique imaging clue. Sometimes doctors see hypertrophy, meaning enlargement, of a structure called the clava in the medulla oblongata, part of the brainstem. That's quite specific when seen with cerebellar atrophy.
So the imaging gives strong clues. The shrinking cerebellum, maybe iron, this clava sign. But the definitive answer comes from genetics.
Exactly. Genetic testing is the gold standard now. Sequencing the PLA2G6 gene to find those two faulty copies, those biallelic pathogenic variants, confirms the diagnosis. It tells us the blueprint for that crucial enzyme is indeed broken. And thankfully, this has largely replaced the need for older, more invasive methods like brain biopsies.
Okay, let's try to tie this together again. The molecular problem, the broken enzyme, creates this, you called it a perfect storm. How does that actually play out inside the neurons? You mentioned oxidative stress and iron.
Right, so the loss of that phospholipase enzyme function triggers essentially a cascade of failure. First, you get major mitochondrial dysfunction. Remember, the enzyme helps maintain mitochondrial membranes and clear out damage. Without it, you get runaway lipid peroxidation fats in the membranes getting damaged by oxidation, and massive oxidative stress builds up inside the cell. This damages the mitochondria, so ATP production, the cell's energy currency just plummets. And those high-demand brain areas like the cerebellum, they're starved for energy.
And then the iron adds fuel to the fire.
Precisely. The iron accumulation that we sometimes see isn't just sitting there, it actively makes things worse. Iron can catalyze a chemical reaction called the Fenton reaction, which dramatically accelerates lipid peroxidation. So think of it like this. The damaged lipids are already piling up like dangerous waste, and the excess iron acts like an accelerant, fanning the flames of oxidative damage. Leading potentially to a specific type of programmed cell death called ferroptosis. It's literally iron-dependent cell death. The neuron is essentially poisoned from within by the toxic byproducts of its own failing structure and metabolism, supercharged by iron.
It's just incredible how one tiny genetic error triggers such a complex, destructive chain reaction. Okay, given how severe this is, let's shift focus. Is there any hope? What's the current state of treatment, and what's the research pipeline look like based on that review?
Well, the current reality is still incredibly tough. There are no approved treatments that can cure INAD or even slow its progression definitively. Management is entirely palliative, focused on supportive care.
Meaning just managing symptoms.
Yes. Trying to control the severe spasticity, often with medications like baclofen, sometimes delivered directly to the spinal fluid. Providing intensive physiotherapy to maintain range of motion and prevent joint contractures. Managing feeding difficulties, often needing feeding tubes. And critically, providing respiratory support as breathing becomes compromised. It's constant intensive care.
The need for something more fundamental is just immense. Okay, the review did highlight some experimental approaches. Since a big part of the problem is that lipid damage, that oxidation, there's one strategy targeting that directly, right?
That's right. There's significant interest in using what are called deuterated polyunsaturated fatty acids, or DPUFAs. Specifically, a compound called RT001 has been explored.
Deuterated. What does that mean? Is it like reinforcing the lipids?
That's actually a great way to put it. Deuterium is a stable, heavy isotope of hydrogen. In DPUFAs, some of the regular hydrogen atoms at specific vulnerable points in the fatty acid chain are replaced with deuterium. This simple swap subtly strengthens the chemical bonds at those points, making the lipids significantly more resistant to being attacked and damaged by oxidation, by that lipid peroxidation process we talked about.
So it's like putting chemical armor on the fats that make up the cell membranes.
Exactly. It slows down that destructive cycle of damage. The hope is that by protecting the lipids, you can mitigate some of the downstream consequences like membrane failure and mitochondrial dysfunction.
And the review mentioned some very early human studies. 2 patients.
Yes, initial open-label studies, meaning not placebo-controlled. The reports indicated the drug, RT001, seemed to be well tolerated. And in one of the 2 children, there were some reported transient, temporary improvements in things like alertness and maybe some fine motor skills in the first few months.
What transient?
Yes, unfortunately, a decline was reported later on. It highlights that while the mechanism protecting lipids is biologically plausible and interesting, it might not be enough on its own to fully halt the disease, perhaps because it doesn't replace all the functions of the missing enzyme.
So more research needed there. Definitely.
Absolutely. Larger controlled trials are essential to really understand if DPUFAs have a meaningful clinical benefit.
What about going straight to the source, fixing the underlying genetic problem?
That's the ultimate dream, of course. Gene therapy is a major focus of research. The main approach being explored is gene replacement.
How does that work?
The idea is to use a harmless virus, usually an adeno-associated virus, or AV, as a delivery vehicle. You engineer the virus to carry a correct functional copy of the PLA2G6 gene. Then you introduce this virus into the patient, aiming for it to infect the relevant cells, particularly in the brain and spinal cord, and deliver the working gene copy.
So the cells can start making the proper enzyme.
That's the goal. Get the cells to produce their own functional phospholipase A2 type Xi. There's also longer-term potential for gene editing technologies like CRISPR-Cas9, which could theoretically correct the specific mutation in the patient's own DNA. But delivering these tools safely and effectively to the brain presents huge challenges still.
And these therapies are developed using models. You mentioned stem cells.
Yes, researchers can take skin or blood cells from patients, reprogram them back into stem cells-induced pluripotent stem cells, or iPSCs, and then differentiate those into neurons in a dish.
So you have patient-specific brain cells to study.
Exactly. These cellular models are invaluable for understanding precisely how the PLA2G6 mutations cause damage at the cellular level and for testing potential drugs, like the DPUFAs or others, before moving to animal models or human trials.
Okay, let's address the big challenge here. We have these promising ideas, protecting lipids, replacing the gene. Why is progress often so slow in getting these from the lab to the children who desperately need them?
The biggest hurdle, honestly, is the rarity of the disease itself. With maybe only 1 affected individual per million births, it's incredibly difficult to conduct large-scale clinical trials.
Not enough patients for a traditional study.
Exactly. You need statistically significant numbers to prove a treatment works and is safe, comparing it against a placebo or standard care. With ultra-rare diseases like INA, finding enough participants, especially for randomized controlled trials, is a monumental challenge. This means that a lot of the current clinical management strategies are based on smaller studies, case reports, or just expert consensus simply because we lack that high-level evidence from big trials.
So collaboration becomes key.
Absolutely crucial. International patient registries, consortia of researchers and clinicians sharing data, that's really the only way forward to pool resources and knowledge for these ultra-rare conditions.
This has been incredibly informative, really painting a clear picture. We've seen that INAD and the broader plan spectrum boils down to this fundamental flaw in the PLA2G6 gene, crippling that vital phospholipase A2 type 50IN enzyme. And the consequences cascade outwards. Failed membrane maintenance, energy crisis in the mitochondria, this toxic buildup involving lipid peroxidation and iron, leading to the devastating neurodegeneration.
That's the core story. And maybe here's a final thought, a provocative connection for you to mull over. That specific destructive pathway we've unpacked in INA, this toxic trio of lipid peroxidation, mitochondrial failure, and iron accumulation interacting, it's not actually unique to this very rare childhood disease.
Oh, where else do we see it?
We see echoes, sometimes strong parallels, of these same molecular mechanisms implicated in much more common, later-onset neurodegenerative diseases. Think about Parkinson's disease, Alzheimer's disease. There's growing evidence that similar processes involving lipid damage, mitochondrial stress, and iron dysregulation play significant roles there too.
Wow. So you're saying that cracking the code for INAD, finding effective ways to target these pathways, whether it's the DPUFAs protecting lipids or gene therapy restoring the enzyme, could potentially have massive implications beyond just this one rare disorder.
It's a really important question, isn't it? Could success in developing targeted therapies for this devastating condition affecting 1 in a million children actually help unlock new strategies, new foundational treatments for neurodegenerative diseases that impact millions and millions of adults worldwide?
That's quite a thought. The fight for the few potentially paving the way for the many.
It's definitely a possibility worth watching very, very closely in the coming years. The research here could have incredibly far-reaching benefits.
Absolutely. Well, thank you so much for walking us through this complex but critical area, and thank you, our listener, for joining us on this deep dive. We hope this exploration provides some valuable insight into the challenges and the hopes on the frontiers of neurodegeneration research.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.